Good afternoon, and thanks to the organizers for this great opportunity to present our results on the role of the mechanosensitive Piezo1 channels on the arterial medial calcification. The vascular calcification is defined as the deposition of the calcium phosphate complexes in large and medium-sized blood vessels. The process is highly correlated with a number of cardiovascular diseases, depending on the organs affected and the degree of progress. Mineral deposition can affect both the intimal and the medial layers of these blood vessels. The intimal layers of the arteries are normally composed of endothelial cells and subendothelial connective tissue. And calcification occurring in this layer is associated with atherosclerotic lesions, with further risk of obstructive vascular diseases, unstable and rupturable plaque formation, myocardial infarction, or stroke. In contrast, calcification of medial layer, which consists of smooth muscle cells and the elastin-rich extracellular matrix, is linked to vessel stiffness, systolic hypertension, and increased pulse wave velocity, leading to increased diastolic dysfunction, and finally heart failure. Medial calcification can occur in a range of conditions, including the genetic disorders, aging, chronic kidney disease, diabetes mellitus, or even hypervitaminosis D. Unlike atherosclerotic calcification, the medial calcification may occur in the absence of lipid accumulation and inflammatory cell infiltration. Originally, calcification was thought to be a passive process. However, it is now known that it is an active and very tightly regulated process, principally driven by the vascular smooth muscle cells. In normal adult tissue, the smooth muscle cells have a contractile phenotype, and they contribute significantly to the physiological function of the cardiovascular system, and also to the maintenance and remodeling of the extracellular matrix. However, unlike other myocytes, the smooth muscle cells are not terminally differentiated and display phenotypic plasticity. Smooth muscle cells can alter their phenotype in response to pathological signals, and they are capable of down-regulating contractile proteins, increasing their proliferation and resulting in the remodeling of the extracellular matrix to facilitate the migration of the cells. Recently, it has become clear that smooth muscle cells are able to maintain a spectrum of phenotypes and can display features of osteoblasts, chondrocytes, adipocytes, and macrophage foam cells as well. The osteochondrogenic transdifferentiation of vascular smooth muscle has a central role in coordinating the processes leading to vascular calcification. In parallel with this phenotype change, calcifying extracellular vesicles deposited and aggregated in the wall of these vessels are also secreted by the smooth muscle cells. Under physiological conditions, these vesicles support the homeostasis and they do not calcify. They contain actually calcification inhibitors as matrix GLA protein and fatwin A. Loss of these inhibitors increases the probability of the appearance of nucleation sites of mineralization. These phenomena resemble bone mineralization, where in growth-based chondrocytes, an intracellular signal has been proposed to initiate transdifferentiation and release of calcifying matrix vesicles. During vascular calcification, the effect of both increased extra or intracellular calcium levels have also been widely studied, and a very clear connection to the calcification have been documented. Several studies have reported the role of the elevated extracellular calcium level in smooth muscle cell differentiation. On the other hand, the mineralization of matrix vesicles has been assigned to disturb intracellular calcium homeostasis. The mechanism, however, whereby this intracellular calcium level is increased in the smooth muscle cells is not properly understood. In 2010, Piezo1 and colleagues uncovered a new group of mammalian mechanically activated channels named the Piezo family. Since the identification of Piezo channels, which won the Nobel Prize, a number of studies have been demonstrated their role in vascular physiology. These pore-forming transmembrane proteins function as mechanosensitive, non-selective cation channels with a preference for calcium. Piezo1 protein has a three-bladed, propeller-shaped homotrimeric architecture. Piezo1 is an unusually large protein containing about 2,500 amino acids, with each subunit with an estimated 2440 transmembrane segments. Their contribution to the altered intracellular calcium homeostasis has been shown to be significant in many cardiovascular diseases. It is known that the Piezo1 channels in the endothelium of blood vessels play an essential role in the early development of the vascular system, and the lack of the channel or a loss of function mutation in the embryonic age is lethal due to the disorganized vascular structure. The expression of Piezo1 is high in the smooth muscle cell of resistance arteries as well. Piezo1 seems to be dispensable for myogenic tone induced by an increase in the wall tension, and it is essential in the arterial remodeling of the arteries during hypertension. Calcium influx through Piezo1 leads to an increase in intracellular calcium concentration. The aim of this work is to demonstrate the essential contribution of the mechanosensitive Piezo1 channels to vascular calcification. Initial investigation tested the relevance of Piezo1 channels in the human vascular smooth muscle cells. Human internal carotid arteries as well as superficial femoral artery samples, both obtained from vascular surgical interventions, have been examined. Immunohistochemistry of these arterial rings revealed that the Piezo1 channel are expressed in the human arteries. Hematoxylin eosin staining has been used to localize the smooth muscle layer in this section, and DAP staining has been applied for the Piezo1 visualization. The fluorescent immunohistochemical staining confirmed this observation on human samples. Here, this red staining refers to the Piezo1 expression, while the green detect the alpha-actinin. Due to the limited availability of these human samples, functional measurements were carried out on the human aortic smooth muscle cell line, the HAOSMC, and partially on smooth muscle cells freshly isolated from most abdominal arteries as well. It's simply to rule out that the observed effects measured on the HAOSMC are attributable to the chosen cell line. After having established that the SMCs, the smooth muscle cells, express this Piezo1 channel, we examine the alteration of its expression upon calcification. To induce calcification, the human aortic smooth muscle cells were cultured in the so-called osteogenic medium, where the general culturing medium was supplemented with 1.5 millimole phosphate and 0.6 millimole calcium for 4 days. After treatment, the Piezo1 mRNA and protein expression were analyzed in both control and calcified cells. At mRNA level, the qPCR, and we used the Western blot at protein level, and we observed a significant upregulation of Piezo1 in calcified cells over controls at both levels. Then the activity of the channel was examined. First, we tested the Piezo1 channels on smooth muscle cells isolated from mouse abdominal arteries. We have loaded the cells with Fura2 ratiometric calcium-sensitive fluorescent dye, and we measured the alteration in the intracellular calcium concentration using the Cooled P340 illuminator mounted on a Zeiss X-ray imager fluorescent microscope. 2 different mechanisms have been used to activate the channels: application of the hypoosmotic extracellular fluid and the administration of the Yoda-1, which is a specific activator of the Piezo1 channels. The hypoosmotic shock as well as the Yoda-1 application were able to evoke calcium transients that you can see here on the above the columns. And then we examined the control and the so-called calcified human aortic smooth muscle cells. Both stimulation, the hypoosmotic shock and the application of the Yoda resulted in significantly greater calcium transients in calcified cells as compared to the controls. Then the addition of 5 micromolar DOCU has happened to the extracellular environment. The DOCU reversibly and selectively antagonizes the Yoda-1 effect, and the application of DOCU lessened the calcium signal substantially, both in control and in calcified cells, pointing out the importance of Piezo1 in regulation of intracellular calcium concentration. We looked for the influence of Piezo1 activation and the consequently elevated intracellular calcium concentration on calcification of HAOSMCs. As you can see, both alizarin staining and calcium measurement of the extracellular matrix show that Yoda-1, so the activation of the Piezo1 channels, largely amplified the osteogenic medium-induced calcification of these cell types. We also measured the level of osteocalcin, the major non-collagenous protein in bone, and a well-established marker of osteogenic differentiation of the smooth muscle cells. The osteogenic medium induced an increase in osteocalcin level compared to control, which increase was further intensified by Yoda. Next, we set up an ex vivo tissue culture model, and we investigated the effect of Yoda-1 on aortic calcification. Cleaned aorta sections of black 6 mice were cultured under control conditions in osteogenic medium and also in osteogenic medium supplemented with Yoda-1. We measured the calcium levels of the aorta rings on day 6, and osteogenic medium, as it was expected, increased the calcium content of the aorta over the control, and the presence of the iota further potentiated the calcium accumulation in the aorta. After 7 days of culturing, we performed an Foncos staining on these specimens to visualize the calcification. The staining revealed no calcification in the control and mild calcification in the osteogenic medium treated cells or cultures, and an extensive calcification when the Yoda-1 has also been applied besides the osteogenic medium. So these experiments also reflect to the involvement of the Piezo1 channel in the medial calcification. We tested DOCU1, the chemical antagonist of Yoda-1 evoked Piezo1 channel activity, just to confirm the role of Piezo1 in the Yoda-1 induced calcification of the HAO-SMCs, the human aortic smooth muscle cells. As revealed by the alizarin red staining and the calcium measurements of the extracellular matrix, the application of DOCU1 at a concentration of 5 micromole inhibited the calcification of vascular smooth muscle cells induced by the osteogenic medium and the supplemented Yoda-1 application. Even the osteogenic medium increased the calcification as compared to general culturing medium, which was potentiated by the Yoda-1, but the DOCU was able to decrease this effect both in alizarin red staining and in the extracellular matrix calcium contained. To further confirm the role of Piezo1 in Yoda-1 induced vascular smooth muscle calcification, we applied an siRNA techniques to downregulate Piezo1 expression. qPCR analysis indicated significantly decreased expression of Piezo1 at mRNA level, both in control cells and also in so-called calcified cells cultured in osteogenic medium supplemented with Yoda. And we have got the same effect on protein level by Western blot, and again, both in case of control cells and in calcified cells as well. Knockdown of Piezo1 by siRNA attenuated the osteogenic medium plus Yoda-1 induced calcification as detected by alizarin red staining, as well as the calcium content of the extracellular matrix. Alizarin red staining in scramble cells indicated a significant increase in osteogenic medium complemented with 10 micromolar Yoda-1 compared to the general culturing medium. And we didn't find similar changes in knockdown cells. The calcium content of the extracellular matrix in scramble cells was also significantly augmented upon induced calcification and by additional stimulation of the Piezo1 by Yoda-1. And however, in the Piezo1 knockdown cell, this effect of the stimulation was totally absent. So we believe that this dramatic suppression of calcification would only be attributed to the lack of Piezo1 channels. And these findings does demonstrate the fundamental role of Piezo1 in calcification. Without sufficient expression or function of the channel, administration of the osteogenic medium and the Piezo1 has no or limited impact on the calcification, as indicated in these two experiments. So we know a lot on smooth muscle cells in vascular calcification, and also the importance of altered intracellular calcium homeostasis, both in smooth muscle cell dysfunction and calcification, is also generally accepted. However, this causal relationship between these two phenomena and the response to how intracellular calcium levels of the smooth muscle cells may increase, leading to vascular calcification, are only partially known. We think that our observations provide one of the missing links, revealing a potential source of intracellular calcium concentration rise in these human aortic smooth muscle cells. Furthermore, they also demonstrate that stimulation of Piezo1 channels significantly accelerates calcification. So altogether, that could mean that Piezo1 channels could play a role in phenotypic changes of vascular smooth muscles. And based on the role of Piezo1 revealed here, they may be a potential therapeutic target that could prevent or at least slow down the progression of arterial medial calcification. I would like to thank to my colleagues, both in the Department of Physiology, László Csernok, László Szabó, Norbert Balog, and Ágnes Angyal, and also in the Clinical Center, Csaba Tóth and Ildikó Balatoni, and also in the Vascular Pathophysiological Working Group. Special thanks to Victoria Jenei and Andrea Todd. And finally, thank you for your kind attention.